guys, welcome to my YouTube channel. So in this video, we're going to talk about the Novo synthesis of purine nucleotides. So remember, there are two pathways for the synthesis of these nucleotides. You have the salvage pathway and the de novo pathway. Now salvage pathway involves the recycling of the nitrogenous bases as a result of the degradation or the catabolism of nucleic acids. Now, the de novo pathway, which is the main pathway, occurs primarily in the liver, and it involves the synthesis of these nucleotides from a particular precursor that we are going to see just immediately. Now, just as we have said, the de novo synthesis occurs inside the liver, and there are particular raw materials that are going to help us in the synthesis of these purine nucleotides. So, let us first of all list the raw materials. So, raw materials. So number one, we are going to have the amino acids. So amino acids. And what are these amino acids? The first one is going to be aspartate. Another one is glutamine. And another one is glycine. Then, apart from the amino acids, we are also going to have carbon dioxide, CO2. Then, the next one, we are going to have phosphoribose. So, phosphoribose. Then, the next one, we are going to have one carbon unit. So, one carbon unit or units, such as the N10 formyl tetrahydrofolate white. So these are the raw materials we're going to use to synthesize these nucleotides. So the aspartate, glutamine, and the glycine are just going to provide the nitrogen needed for the synthesis of these nucleotides. Carbon dioxide is going to provide the carbon. Now let us see the sources of the nitrogen and carbon for the purine nucleotides. So sources of carbon and nitrogen. So the first one is aspartate. Aspartate is going to give us the nitrogen number one. Then CO2 is going to give us carbon number 6. Right. Then glutamine is going to give us nitrogen number 3 and nitrogen number 9. Then we have what? We have glycine. Glycine is going to give us carbon number 4, carbon number 5, and nitrogen number 7. Then we have the one carbon unit, the N10 formyl tetrahydrofolate. The N10 formyl tetrahydrofolate. This is going to give us carbon number 2 and carbon number 8. Alright. Now we have the raw materials and sources of carbon and nitrogen we need for the synthesis of the nucleotides. Now, let us get to that. Hope you guys remember the hexose monophosphate pathway from biochemistry. The hexose monophosphate pathway, the HMP. In the HMP, you remember at some point when we had the ribulose 5-phosphate. So, ribulose 5-phosphate, and we said that ribulose 5-phosphate can be acted upon by two enzymes. Now, when it is acted upon by an isomerase, that is ribulose 5-phosphate isomerase, let me just write here isomerase. Let me set it for the pen. When it is acted upon by an isomerase, you are going to get ribose 5-phosphate. So, ribose 5-phosphate. Then, when it is acted upon by an epimerase, that is 3 epimerase, so It is called the ribulose 5-phosphate epimelase. So let me just write epimelase. We are going to get a molecule called xylulose 5-phosphate. Alright. Now it is this ribulose 5-phosphate that we are going to use as the initial, the intermediate for the synthesis of the purine nucleotides. So let us take this ribulose 5-phosphate. Start here. So you have the ribulose 5-phosphate. Now, ribulose 5-phosphate can be acted upon by an enzyme. Now, what is the name of this enzyme? Ribulose 5-phosphate can be acted upon by an enzyme, and this enzyme is called PRPP Syntheis. PRPP in full simply means 5-phosphoriboside 1-pyrophosphate. Right. So we are going to form a new molecule called PRPP. Let me just write it in full. 5-phosphoriboside 1-pyrophosphate. Alright. Now, this reaction requires some energy. Some energy in form of ATP. So we're going to have ATP, then we convert it to AMP plus pyrophosphate. Alright. We are moving on to the next step. The 5-phosphoriboside pyrophosphate, also known as PRPP, can be converted into a new molecule. This new molecule is called 5-phosphoriboside amine. It's called 5-phosphoriboside amine. 5-phosphoriboside amine. Alright, or you can just call it PRA. 5-phosphoriboside amine. Now, this reaction requires an amino group, just as you can see here, provided by glutamine. Glutamine is going to be converted into glutamate. As you know, the difference between glutamine and glutamine is only the amino group. That's glutamate has 2 amino groups, while glutamate has 1. So it has lost the amino group to PRPP from PRA. Alright. Now the enzyme that catalyzes this reaction is called PRPP glutamine aminotransferase. Alright, PRPP glutamine aminotransferase. And guys, this is the committed step in this particular pathway. So I'll use this is the committed step. Alright, let's move. 5-phosphoriboside amine is going to be converted into a new molecule. Now, this reaction is going to require glycine, amino acid glycine. So we're going to have glycine plus ATP. We're going to have ADP. Alright. Now, what is the name of the molecule that's going to be formed? It's going to be called glycinamide riboside 5-phosphate, or glycinamide ribonucleotide. So glycinamide riboside 5-phosphate. Alright. What is the name of the enzyme that catalyzes this reaction? This reaction is catalyzed by an enzyme called glycinamide synthetase. So glycinamide synthetase. Alright, guys, so let's move. Glycinamide is going to be converted into a new molecule. Now, this glycinamide is going to require N10-formyl tetrahydrofolate. So we're going to have N10-formyl tetrahydrofolate. Then you are going to get rid of the tetrahydrofolate. Alright. Then we are going to form a new molecule, and this new molecule is going to be called formyl glycinamide ribosulfite phosphatol. If you like, you can call it ribonucleotide. So ribosyl 5 phosphate. Alright. Now this reaction is catalyzed by an enzyme called GAR transformylase. So transformylase. GAR simply means glycinamide and ribosyl phosphate. Alright. Now let's proceed. Formyl glycinamide ribosyl phosphate going to be converted into a new molecule, and this new molecule is going to require glutamine amino acid. So we're going to have glutamine. It's going to be converted into glutamate. So to me, the glutamate is going to deliver its amino group. Now we're going to form a new molecule, and this new molecule is going to be called formyl glycinamide ribosulfite phosphate. So ribosyl 5 phosphate. And this reaction is going to be catalyzed by an enzyme called Syntheis enzyme. It's going to take the amino group from glutamine, add it to formyl glycinamide ribosulfite phosphate from formyl glycinamide ribosyl 5 phosphate. Alright, let's move this side. So here we are formyl that one is going to be converted to a new molecule. So let us see this. Here we are formyl glycinamide ribosulfite phosphate. So formyl glycinamide ribosulfite phosphate. Ribosyl 5 phosphate. Alright. Now it's going to be converted into a new molecule. So this one is going to be converted into a new molecule. And this reaction is going to require ATP. So I'm going to input some ATP energy in the form of ATP. ATP is going to be converted into ADP plus phosphate group. Now, what is the new molecule? Now from here, we are beginning to form the ring of the purine. So this we are going to form a molecule called imidazole. Actually, it's 5 imidazole. Imidazole ribosulfite phosphate. Ribosyl 5 phosphate. Now, this reaction is going to be catalyzed by an enzyme called Syntheis. So, Syntheis. Alright. Then from 5 imidazole ribosulfite phosphate, we are going to a new molecule, and this new molecule is going to be called imidazole carboxylate ribosulfite phosphate. So let me write this in the calculator. So imidazole carboxylate ribosulfite phosphate. Ribosyl 5 phosphate. And this reaction is going to involve the input of CO2. So we are going to input CO2. Or you can just, sometimes you can see it is the bicarbonate ion that is added. So don't be surprised. So this reaction is going to be catalyzed by an enzyme called carboxylase. So carboxylase. as the name suggests, carboxylate is just going to carboxylate, add the CO2. Now, moving down, we're going to have a new molecule, and this new molecule is going to involve an input of amino acid aspartate. So, we're going to add aspartate. So, aspartate. Alright, now when we add aspartate, we are going to form a new molecule called aminoimidazole. Aminoimidazole 4-succinyl carboxamide ribosyl 5-phosphate. Alright. Aminoimidazole 4-succinyl carboxamide ribosyl 5-phosphate. Some foods you can be written as succinyl aminoimidazole 4-carboxamide ribosyl 5-phosphate. So don't be surprised when you see that. Alright. This reaction is going to be catalyzed by an enzyme called Syntheis. Alright. Then moving down, aminoimidazole 4-succinyl carboxamide ribosyl 5-phosphate is going to be converted into a new molecule. So actually we are going to have a cleavage. So we are going to release fumarate. So I'm going to release fumarate. So to me we are going to form a new molecule, and this new molecule is going to be called aminoimidazole 4-carboxamide ribosyl 5-phosphate. Ribosyl 5-phosphate. Alright, so the name of the enzyme that catalyzes this reaction is going to be called adenosaccinate lyase. It's a lyase. It's going to lyase the aminoimidazole 4-succinyl carboxamide ribosyl 5-phosphate to release the fumarate. And of course, you know where we can take this is a component of the TCA, right? So we are going to form aminoimidazole 4-carboxamide ribosyl 5-phosphate. Then, moving down, we are going to convert this into a new molecule, and this new molecule is going to be called it's going to involve the addition of a formyl group, and the formyl group you obtain from the N10 formyl tetrahydrofolate. N10 formyl tetrahydrofolate. We are going to release the tetrahydrofolate. Alright. Then we are going to form this new molecule. It's going to be called aminoimidazole 4-carboxamide ribosyl 5-phosphate. Ribosyl 5-phosphate. Alright, this reaction is going to be catalyzed by an enzyme called formyl transferase. So, formyl transferase. So we're going to have formyl transferase. It's going to transfer the formyl group from the N10 formyl tetrahydrofolate into aminoimidazole 4-carboxamide ribosyl 5-phosphate to form aminoimidazole 4-carboxamide ribosyl 5-phosphate. Alright. Then from there, we are moving down from a new molecule, and this new molecule is going to involve the release of our water molecule. So I'm going to get rid of our water molecule. So this reaction is going to be catalyzed by an enzyme called cyclohydrolase. We're going to have cyclohydrolase. Alright, then what are you going to form? You're finally going to form the inosine monophosphate. Also abbreviated as IMMP. So those are the steps of the de novo purine synthesis, and this acts as the parent for the synthesis of other purine nucleotides, the inosine monophosphate. So we all started from the ribose 5-phosphate, we went down through the steps, including the enzymes and everything, and we finally come at the inosine 5-phosphate. So we want to see how we can mobilize this inosine 5-phosphate to generate AMP, GMP, right? Now, let us come back here and see. Let us see what is going to happen. The inosine 5-phosphate is the parent from which you are going to synthesize other nucleotides. Now, when you have inosine monophosphate, now we can take this inosine monophosphate and react it with a new molecule called aspartate. So we take IMP, react it with aspartate. We're going to form a new molecule. So when we react them, we're going to form a new molecule called adenyl succinate. It's called adenyl succinate. So this reaction requires energy in form of GTP. So we're going to have GTP, we're going to be converted to GDP. Alright. Now, adenyl succinate can be cleaved by an enzyme called adenyl succinate lyase. I told you the lyase work is to lyse or to cleave. So adenyl succinate lyase. Alright, then adenyl succinate lyase is going to lyse the adenyl succinate, and we are going to form adenosine phosphate. You know this? This is a nucleotide. Of course, I'm sure you encountered this before. The adenosine monophosphate. This is the synthesis of purine nucleotides. Of course, no, adenine is a purine. So that's why this is a purine nucleotide. Now let us also see how we are going to synthesize the GMP. So we can take inosine monophosphate. Let me just show it that way. So you can take IMP. IMP is going to be acted upon by an enzyme that is going to involve conversion of NAD+. Let me use a different pen. I'm going to have the NAD+. NAD+ is going to be converted into NADH. It's going to be reduced. Now when it is reduced, you are going to oxidize the inosine monophosphate. So when you oxidize the inosine monophosphate, you are going to get an enzyme, a new molecule called synthesis monophosphate. Synthesis monophosphate. Now this reaction is going to be catalyzed by an enzyme. Now this enzyme is called IMP dehydrogenase. Now I told you that whenever you see a dehydrogenase, it means you are going to involve the reducing agents. Right? Now the synthesis monophosphate can be converted into guanosine monophosphate, right? And this reaction is going to involve addition of a nitrogen, right? Or the amino group from glutamine. It's going to be converted into glutamate. Alright. Now we finally have our GMP and our AMP. Now, you know that nucleotides are sources of energy. They can provide energy in metabolic transactions. So to me, you can further convert these, or in a way convert them into ATP. That's pretty cool. You can convert also this one into ATP because when we break it down, we are going to release the phosphate groups, and no phosphate groups contain energy. Alright, so that is all that we have for the Novo synthesis, but also we can also talk about the regulation of this pathway. So, regulation. So, in terms of regulation, you can say that after we synthesize enough adenosine monophosphate, this adenosine monophosphate can in turn inhibit the adenine succinylase, right? So, they can inhibit. This can be inhibited by AMP. Alright. Then also when you have a lot of the guanosine monophosphate in our body, we are going to have the negative inhibition or negative feedback on this particular enzyme here. So we are going to be inhibited by GMP. When you have elevated amounts of GMP. Alright. We have another enzyme called 6-mecaptopurine. 6-mecaptopurine is going to inhibit the synthesis of both of these. So it's going to inhibit the conversion of inosine, any other reaction beyond the inosine monophosphate. So we're going to have this pathway is going to be inhibited by 6-mecaptopurine. 6-mecaptopurine. Alright, then let's again come here. You know this PRPP synthesis, which was converting the ribose 5-phosphate into 5-phospholipose 1-5-phosphate, or in short, the PRPP. This one can be inhibited by all of these three, the ALP, IMP, the GMP. So can be inhibited by GMP, can be inhibited by IMP, and also be inhibited by AMP. So all these are going to inhibit the PRPP synthesis, so that you cannot convert the ribose 5-phosphate into PRPP. I mean, maybe you have a lot of nucleotides in our body, yeah. Alright. The PRPP synthesis can also activate this particular enzyme, the PRPP glutamyl aminotransferase. So you can stimulate, because remember I told you this is a committed step in this particular pathway. That's why I used this thick line to show it's a committed step. If I can let you a committed step. Committed step. Alright. Then so I've said the PRPP synthesis can activate PRPP synthesis can activate the PRPP glutamyl aminotransferase. So guys, that is what we have about the Novo synthesis pathway. So in another video, we are going to see how you can synthesize the nucleotides from the using the salvage pathway. Just as I told you, the salvage pathway is the pathway in which we recycle the nucleotides, so the bases that has been broken down as a result of the nucleic acid metabolism. So you remember, we also can break down the amino acids, the nucleic acids to form the petro sugar, the protein and the phosphate. Can break down those components into individual components. So we can now take those the bases, then you can run them through the salvage pathway to form these nucleotides, so that you can make them available for our body. And actually, a large amount of the nitrogenous bases that have been formed from the catabolism of nucleic acid are not converted to uric acid and maybe nucleated and maybe passed away. So it's just the salvage pathway, and we're going to see that in our next video. So guys, that is it for this video. If you found this video helpful, please subscribe to my YouTube channel, hit that like button and comment.